Okay, hey guys. This is our next part of the video. We finished with what's organic molecules contain, where I said they contain carbon and hydrogen. Our next one is what are defined isomers. Now, isomers are really cool. I love thinking of isomers as Legos, right? Let's say you get a bunch of Lego bricks. You know what? I'm gonna change these to different colors. Right? Let's say we get these 3 pieces of Lego bricks, right? You can make multiple combinations out of them. You can make them into a straight line. You can make them stacked upon each other. You can make them stacked under each other, okay? But they're still made of the core concept. They're made of the same 3 pieces. That's what isomers are. Let's say we had a molecule that was HCH. I don't think that's molecule accurate, but that's not the main point here. Right, so let's say this is our molecule. We can still, actually, let's make it like H, like this. Right, we can change it up to make it HCH with the H right here. We can make it to H to an H with the C right here, something. Now, mind you, these are not molecularly accurate. It's just a representation. And you can just change it up. Isomers are when you take atoms and molecules and just rearrange them. They're the same core components, but you rearrange them. And when you rearrange them, they can have different properties. For example, methamphetamine, which is a famous drug, also known as crack in the hood areas, is a very famous drug for its addictive properties. But their isomers is literally methamphetamine, but just flipped. Let's say the methamphetamine looked like this, right? It just flipped it to like this, right? The fact that it flipped makes it into an over-the-counter drug, which is used for headaches. That's literally it, right? Something as simple as just flipping the shape, rotating it, changing one atom to a different atom, literally can change the entire function of the atom itself and its the entire function of the molecule itself and its purpose. So that's what isomers are, just the changing of the molecule rearrangement and its functions changed because of it. Okay, next part is base pairing with DNA. You guys should know this by now. This should be ingrained with you, okay? Remember, A to T, C to G, that's for DNA. DNA only has T, which is replaced with uracil in RNA. If you want a much detailed video, I highly recommend watching the DNA video that I have made. I am not going to go over this. This is so easy, right? Same with the monomers of DNA and its parts. Please just watch the video. I don't want to really go over it right now when there's steps like explaining meiosis and mitosis, which require 2 times more of the effort. So, you know what? You guys can just watch those videos. I'll link them in the description below. You can just check out my channel as well. Like, really not that hard, right? Now, let's go to the big guns here, okay? Steps of mitosis and meiosis. Let's go with mitosis first because that is the easiest one. Okay? Let's do this, guys. Remember, it's in PMAT, right? PMAT. Prophase. Metaphase, anaphase, telophase. Right? These are the main parts. So let's look into it. You know what? Let's look into it, you guys. Right? We're not going to look at meiosis. We're looking at mitosis. Remember that. Right? Right. Alright, I'm not going to draw every single part because that's going to take way too much time. So, I'm going to be the bigger man here and just search it up. Hey, work smart, not hard. And I'll come by and explain every single topic. Yep, that's a good image. Alright, this is mitosis. Okay, so how does mitosis work? First, we are in the normal phase, right? Now, there is another part called the interphase. That's just very before the actual mitosis happens. You don't even memorize that. Remember just these 4 terms. Okay, these pro-metaphase and all that stuff, you don't even know. Okay, first, Let's look at the cell. It has 4 chromosomes, right? And they look perfectly fine, right? They're a little squished around, really fine, right? And there's these meiotic spindles. These are the centrosomes, right? These will help pull the chromosomes in. You'll find this out later, right? Let's first look into this. There are 4 chromosomes here in the nucleus, right? And there's these centrosome spindles that are around. These will help pull the chromosomes out of each other. I wish you can see my hands as I'm doing this because it would look so helpful. But it's pulling. It's gonna pull the chromatids. Let's look at some basic vocab before we start any further. Right. This example. This is gonna be our DNA. Right. The lines itself. So let's. I like the diagram where it looks like this. Right. Where it looks like this. Each of these lines are called These are chromatids. Right. Oh my god. Chromatins, right? That's what each of the lines are. Each of these are 1 chromatin. So there's 2 chromatins. And these spindles will pull the chromosomes and rip the chromatins apart. Now let's look at the second part. See, now they took that nucleus and put it in the center. And they're setting up each of those chromosomes in line so that they can be ready to split them apart. See how they're trying to grab each of those chromosomes, right? So that the spindles are trying to grab each of those chromosomes so they can start ripping it apart. Next, we're going to look at metaphase. Now the chromosomes are all lined up in the metaphase plate. Metaphase plate, right? They're all lined up and the spindles are ready to pull as well. Then the spindles take it and they start pulling each and every single one of them, right? Pulling each and every single one of them, separating them. Then they split, right? They split with their own cells. And what happens next is that DNA replication fills in the rest of the chromosomes, making it perfectly 2 identical daughter cells. That's the entirety of mitosis. How difficult was that? Explain to me. That wasn't hard at all, right? And, the cell fills up the rest of the part. They make the new cell memory. They make the extra cytoplasm to make everything work over again, right? Remember the metaphase plate. Remember all those main vocabulary words. But in general, it wasn't that difficult. Remember, this is a normal cell. There's no 46 chromosomes. There's only 23 chromosomes. Okay? That's the entirety of it. There are 23 chromosomes, first and final. Okay? These centromeres take their fibers, their spindle fibers, and rips the chromosomes apart. Okay, that's the entirety of mitosis. I don't think we need to go any more detailed. Right? No, we don't. Oh yeah. They say 46 chromosomes. Okay, my bad. I was looking at this differently. This is, when they talk about 46 chromosomes, they're thinking of this as a duplicated chromatin. Okay, so what happens in, interphase is that they duplicate the amount of chromosomes in it. My bad. So you know how it says 46 chromosomes here? The amount of chromosomes that's already there is 23. They duplicate it fully, so that's 46. So that when they split, it's a normal amount on both sides, right? 2 daughter cells, 46 chromosomes each. That's supposed to be right. No, am I looking at this is mitosis or meiosis? Oh, okay. I am so sorry. So the image that I'm using is a mix of the two, right? It's like this weird. The way I found this image is like a mix of both mitosis and meiosis. It's like kind of gray area. It's not good, but there's not 46. It's just it's 40. It's 26 chromosomes, and they eat. There's 46 chromatins, not chromosomes. It's supposed to be chromatin. Then what did I say? What chromatins were? There were those lines, right? And then they split them until there's 26 lines, and then there's 26, and those become there's 23 chromatins in each cell, which then turn into 23 full chromosomes, because one cell has 23 chromosomes. Okay. Oh, shucks. Whoa, wait. I'm wrong. I'm completely wrong. I am stupid. Okay. There were 46 chromosomes in total, right? There are 46 chromosomes in total. Remember, chromosomes are the 2 on 2. There's 46 chromosomes from total. Okay, with 23 pairs of chromosomes. So, these lines in self, called chromatins, are also called chromosomes, right? Right? Chromatins Okay? They're the same word for chromosomes. There are 46 total chromosomes. So if they ever say chromosomes, think of chromatins. I am so sorry. If you guys want me to re-explain this in the VC, I'll do it. Okay, next topic, meiosis. This is definitely the hardest one. Okay, there are some form factors that you need to understand before we actually fall into meiosis. Okay. Yeah, this is the perfect image. Damn it. Actually, that's a bad word to say. Alright, perfect. Alright. This is the entirety of it. Alright. Okay, let's look at it. Okay. Prophase 1, metaphase 1, anaphase 1, telophase 1, and then the yada yada yada. Okay. Meiosis, okay. The second version of meiosis 2, is very eerily similar to mitosis, right? Remember, there are 2 full sets of chromosomes. 1 full set from the mom, 1 full set from the dad. What does that mean? There are 23 pairs. 23 pairs of chromosomes from the dad and 23 pairs from the mom. So in total, there's 46 pairs. There's 46 pairs. 46 pairs. Okay? Right? And then they do the exact same thing. Right? So let's look into each step very closely. Right? They have the cell open. They're just like regular. And then metaphase 1. We're going to skip it directly. Right? We're not going to look at that thingy-majiggy. They set ready in the metaphase plate. Remember the metaphase plate? Now, during the metaphase plate, these chromosomes are so next to each other. 1 from the mom, 1 from the dad, 1 from the mom, 1 from the dad. And what they do is something called crossing over, where because they're so close, they actually share some genes together and mix it up. That's why you see a small red section here and small blue section here. They're sharing some of their information together, which causes them to have crossing over. This allows for less genetic likeness, and creates more genetic diversity, which allows for the less likeliness of disease, mutations, and other complications during pregnancy for the child, right? So these crossing overs are really, really important. Okay, remember, metaphase plate, there's crossing over. I will review this in the VC. Don't worry about it. Then the meiotic spindles come and they pull it, right? And actually, what happens is there's 2 full pairs of chromosomes. So it's 23 pairs here. 23 pairs after the end, right? Now, those 23 pairs of chromosomes then get split into 23 chromosomes in total. 23 chromosomes in total, which means 23 of these stick thingy-majiggies, right? There's 23 in each of them. And then they get split normally as in mitosis and make the normal cells. Wait, that wasn't really hard to explain. If you guys want me to explain this all over again, I will in the VC. But this is really the entirety of it. I think I'm overcomplicating the issues of it. Prophase 1, cells are there. They set up in the metaphase 1. They share some genes together. That's called crossing over. They split like normal. There's 23 pairs in each of them. Then in meiosis 1, each of those 23 pairs gets split again to 23 normal chromosomes or chromatins per cell, which are then split just like meiosis 1. Remember, they're in the metaphase plate. They get split, right? And when they get split, right, when they get split, they just do the normal thing, right? So they're 23 pairs. Oh, it's going this way, by the way. My bad. But I still explain it fine. It's going this way, right? But that's the whole issue, the whole thing. Now, define ocean acidification. That's our next topic. You know what? I'm just going to finish the entire thing. Okay. First, define ocean acidification. She should have put this back in the top with the climate change and everything. I can't really show an image for this. Actually, I'm going to pull up an image. You read it, but I'm going to explain it in my own terms while you see the image, okay? Ocean acidification. It's a really good one. Yep. You guys can look at this. Shucks. What the hell? Y'all can look at this, right? As I try to explain it. here's a better, here's one with information in it as well. Right, okay, so you guys can read these images while I try to explain it with my own words. Think of it like a podcast moment. Okay, due to CO2 created by cars, gases, and just from us, right, there's not enough photosynthesis to deplete all that CO2. And also, the atmosphere isn't strong enough to release all that CO2 as well. So all that CO2 is just muddled there in the air. So what happens next? Instead of just staying there in the air, making it just more and more toxic for us human beings in the land, they start to fuse with the water because they can easily connect through their molecule systematics. The CO2 coming in dissolves with the ocean and reacts with H2O and makes carbonic acid as a return. Now, since it's an acid, it acidifies the water, lowering the pH level, making it more acidic, which is lethal for organisms because they're not adapted to it. Carbonate, seawater acidifies and HI ions, which are the parts of the factors in pH, right? Combine with the carbonate ions to create bicarbonate, right? So there's H ions from the pH scale. Remember when we talked about those? Because it becomes acidified, they fuse with that carbonate ions, making bicarbonate. Those carbonate is required for calcification by many marine organisms, including reef-building corals. Less carbonate is available for calcification. So because of that, carbonate ions are fused with the H ions to make bicarbonate. Those carbonate ions are not there anymore, right? And those carbonate ions are actually needed to create coral reefs. And since there's no more of it left, the corals can't create their coral reefs, and it causes so much issues for marine life. That's the general sense. I don't think we can get any simpler with that. You need me to explain it? Again, I will. Just ask me in homeroom. But I'm not trying to I also have a life. I want to play my Minecraft. Okay? So, that's the entirety of it. Next, essential dogma of each sector. Dogma. Okay, it's just translation and transcription. I, first of all, explained in extreme detail transcription. Right? I know for a fact that I explained that. Translation and transcription, I explained that in my YouTube channel. Right? Yeah, no. You have transcription and translation, I both explained. It was like 17 minutes long. You guys watch that video. Okay? Watch the video. I'll link all those videos in the actual video. Okay? That's everything. That's actually everything, you guys. You shouldn't be worrying about it at all. You guys are done. Okay? Videos on DNA replication, videos on this DNA itself, videos on the central dogma, translation, transcription. I have each and every single one of those. I will link them in the description and you read those. Okay? Those are also going to be useful. I physically cannot make myself to work any harder. I'm done. I need to make my Google video so that I can explain to kids who need questions. So I hope you guys understand. If you have any questions, talk to me in homeroom. You guys have the time. Email me. I can easily answer. Okay. Also, seating chart. My bad. Seating chart. I sit Hirak next to me and Vinay next to me. Actually, there's some flaps. I think Rayan has a proposal to Vinay. Talk about it later. But Rayan's gonna sit next to me. Diagonally can either be Muhammad or Tanush, and diagonally other can be someone else. Just so you know, don't sit behind me. I'm too big, and don't sit in front of me. You're not gonna ever see my screen, and you're gonna keep on turning your neck around. You're gonna be noticed. Other than that, yep, that's everything. Have a great day, guys. And I will make the video. I'll make the DC group chat at exactly 8:30. Get ready, 8:30 EST. See you guys. Have a great day.